Good morning, student. Now here we are dealing with meiosis. As in our previous class we were dealing with mitosis. Today we are dealing with meiosis. Now basically what is the difference between meiosis? See, they take place only in the sexually reproducing cells to form gametes. That means only cells that is present in the sexual organs of both plants and animals, they form gametes. Okay, and generally it is also a very common feature for sexually reproducing. That means we don't find them in asexually reproducing cells. So gametes. Now when the gamete is formed, the chromosome number is haploid. That means half. So that is the reason, since the chromosome number is reduced, it is called reductional division, and in mitosis that is known as equational division. Now you see the phases that we have done in mitosis, same is that of the meiosis, but here meiosis takes place in two phases rather. It is meiosis 1 and meiosis 2, and only a single cycle of DNA replication takes place. Okay, now what happened? Chromosomal mixing between two non-sister chromatids are there. So these are the two distinct features. That means what? That DNA replication completes in the first meiosis 1. Okay, so meiosis 1, in meiosis 1, the prophase 1 is supposed to be a very long stage. So what are these? See, one is liptotene, zygotene, packetine and diplotin. So prophase 1 is same as that of the mitosis. You see, in liptotene what happens? Compaction of the chromosomes. That means chromosomes becoming condensed and visible. Okay. Now you see, chromosome in zygotene, they start pairing. That means what? Similar type of chromosome starts pairing. That means we call it that homologous chromosome starts pairing. And in that they form a connection. The cell, for example, this is one cell, and here also it forms a cell. Like, see, these are the tetrad formation, and here the connection is formed. So this connection here is known as synaptonymal complex. That means they come together, and in the later part, see, in the packetine stage, you see 4 chromatid, 1, 2, 3, 4. This tetrad formation is very distinct, and this is also known as a chiasma formation, and where the crossing over. That means genetical mixing. For example, this is a kind of a one, and this is here. Okay, I'll use a different color code. For example, this has okay. Now after crossing over, we have here. That means what? Part of it has come here. Okay, so this is known as a chromosomal mixing. Okay, and after that, when it goes to the diplotin stage, this synaptonymal complex, they dissolve. That means they come apart. Okay, and then follows the diakinesis. Diakinesis means nucleus start dividing, and we have metaphase 1, anaphase 1, telophase 1, exactly like that of before. Now here you see this prophase 1. See, again same thing, that this crossing over has taken place, and here you see again the metaphase, they keep aligned on the equator. Anaphasic movements are there, and then again you go to telophase. See, this is the last part, and this cell, see this cell, this is again prophase 2, that means meiosis 2. You see the chromosomal duplication does not take place. Okay, so now it goes like this, it is aligned here, again it aligned here, and ultimately it goes like that. So here you see, first we had 4 number of chromosome, and here we have 4 sister chromatid. So this is very important. Okay, you see this line over here. See, 4 haploid daughter cells are formed, 4 haploid. So that is their cytokinesis. So this is the telophase 2. So what is the significance of meiosis C? Significance is, see, the main significance here is it brings about genetical variation. See the word variation. Okay, genetical variation, and this is very important for the process of evolution. Okay, so you have to go through it in detail.